Hello, everyone, and welcome to today's webinar, ASBMB Reciprocal Regulation between Fibrinolysis and Lipid Metabolism. I'm Chenmin Jiang from Publications Department at ASBMB, and I'm thrilled to be your moderator today. This webinar is part of our Breakthrough webinar series, sponsored by the ASBMB journals. Specifically, today's session is brought to you by the Journal of Lipid Research, one of our esteemed ASBMB journals, which has been at the forefront of lipidomics and metabolism research for over 60 years. Before we dive in, a quick reminder, you are welcome to submit questions at any time using the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. This webinar is being recorded, and the recording will be available on the ASBMB website. It's now my pleasure to introduce today's speaker, Dr. Zheng. Dr. Zheng is a junior associate editor of ASBMB Journal of Lipid Research. She started her research lab at the Medical College of Wisconsin and the Versiti Blood Research Institute in July 2020 to study the roles of hepatocyte-derived fibrinolytic enzyme in lipid protein metabolism in atherosclerosis and thrombosis. She is passionate about blood research because blood flows through every organ in the body and it's vital to all human diseases. Without further ado, please join me in welcoming Dr. Zheng. Dr. The floor is yours. Thank you.
Thank you, Chenmin, for the nice introduction, and I really appreciate the opportunity to share my study with this larger audience through ASBMB. Now I'm going to share my presentation. Right, so hopefully everyone can see it now. And so the title of my talk today is the reciprocal regulation between fibrinolysis and lipid metabolism. My research, most of the study that I will be talking about are based at my institution in Milwaukee at the Medical College of Wisconsin and also the Versiti Blood Research Institute. And so to begin my talk, I want to first introduce the concept of fibrinolysis. So fibrinolysis, if you're just looking at the word, is the lysis of the fibrin. So what is the fibrin? Fibrin is the end product of blood coagulation and generate this cross-linked fibrin molecule, which accumulates on the injured lumen of the vessel. And with the accumulation of the fibrin clot, and that will lead to the growing of a blood clot and will eventually block the blood flow and leading to tissue ischemia, for example, in heart attack or stroke. And also venous thrombosis. So with this accumulation of the fibrin on the injured vessel, and the TPA is thought to be released from the endothelial next to this injury site, and TPA stands for tissue plasminogen activator. And TPA can bind to the cross-linked fibrin molecule, where it converts the zymogen plasminogen to the active enzyme as plasmin. And plasmin is the serine protease that can degrade the cross-linked fibrin molecule into the fibrin degradation products. So although the field has been well recognized, the potential of release large amount of TPA from the endothelial cells next to the injury site upon vascular injury. However, this theory does not fully explain how does the large vessels can respond to the thrombotic injury, because if you look at the distribution of the TPA expression on the different regions of endothelial bed, it actually have lower expression in the larger vessel endothelium. And also the large vessel, if you do a math for the surface to volume ratio, it actually have a much lower ratio for the surface to volume, and they also have a much more rapid blood flow. So taking together with all of those points, if a large vessel has a thrombosis occurs, although the local regions endothelial cells can release TPA at some level, but it perhaps not really enough to dissolve the large thrombi accumulated in the region due to the surface to volume ratio that is lower, and also have overall lower TPA expression, and also the rapid blood flow will just flush it away very quickly. So then the question is, how does our body really dissolve some of the larger thrombi accumulated? And also there are evidence showing non-vascular injury induced circulating TPA at the basal conditions. And also there are functions of TPA that not really directly associated with its role in dissolving fibrin clot. So all of those questions leading to one new question is where the basal TPA are derived from. So with this question, we went through the older literatures, and then we found that although there are many cells has been described for TPA expression, but not really been observed in hepatocyte before. However, a study carried out by Dr. Wendy Mars about almost 30 years ago now showed that when she using a monoclonal antibody to inhibit the UPA, which is another serine protease similar to TPA, in the primary rat hepatocyte, she still observed residual plasminogen activation activity when the UPA are completely inhibited. And she had the hypothesis that this perhaps are TPA in the hepatocyte. So to further elucidate this hypothesis, we used a AAV model to silence the TPA expression in the hepatocyte in the mouse model, where we achieved a very significant silencing of the hepatocyte expressed TPA, and without interruption for the endothelial TPA levels. And with this model, we continue to challenge the mice with the thrombosis injury triggered by the ferric chloride applied to the mouse carotid artery, and then we monitor the blood flow to see how quick the thrombi will be formed or dissolved. So within this model, we saw that although the hepatocyte silenced the TPA mouse or having the lower basal TPA concentration, however, it does not impact the mice to continue release more TPA, and perhaps from the endothelial cells after this thrombosis injury. So with this experiment, we conclude that in addition to the known source of TPA in circulation that perhaps derived from the endothelial cells after acute vascular injury, we found hepatocyte can constitutively synthesize and release TPA into the circulation and contribute to the basal fibrinolysis before the thrombi occurs. So the next study, we want to figure out more into the biochemistry, because as a serine protease, we would expect that if there are more protein, its own enzymatic activity should be also higher. However, this is not really the case for TPA. Because if you look at the human plasma, there is a very odd negative correlation between TPA enzymatic activity to its own antigen levels. So how this could be. And the PI1 or the plasminogen activator inhibitor 1 is the major inhibitor of TPA. And if you look at the human plasma, it negatively correlates with TPA enzymatic activity, which makes sense because PI1 is the inhibitor of TPA. However, what's really interesting is that the PI1 antigen level also positively correlate with the TPA antigen in human plasma. And so we generate a hypothesis that whether the high level of PI1 actually can serve as a compensatory stimuli to the TPA levels. And to answer this question, we did a very simple experiment at the beginning, just to inject the recombinant PI1 into the mice, the lean mice that have basal low level of TPA and also PI1. And shortly after 1.5 hours, this recombinant PI1 injection stimulated the PI1 TPA, increased the liver TPA mRNA levels. And also we compare the expression for both PI1 and TPA in the lean versus obese mice, and both are higher. However, the level of the fold increase of PI1 in obesity is much higher than at 12-fold increase compared to the smaller fold increase of TPA in the obese mouse liver, and which also showed reduced liver TPA enzymatic activity in the obese livers. So to really show whether the PI1 can serve as a stimuli to TPA levels in a causative way, we used another model of the hepatocyte PI1 specific knockout obese mice, because the idea is that if we take away the stimuli, which is PI1, do we still see the higher TPA levels that we observed in the obese mouse livers? And using this mouse model, after knocking out the PI1 in the hepatocyte, we observed a 70 reduction of the plasma PI1 antigen levels with an increase of TPA activity and also fibrinolysis potentials measured by the euglobin clot lysis time. And all of those are expected because we are reducing the major inhibitor of TPA. So we see the increase of its activity and also fibrinolysis. However, what's really exciting is that we see this reduction of the TPA antigen levels. Even this is in the obese mouse. So this means that PI1 level can really serve as a stimulation to the plasma TPA levels. So in summary of these studies, we found that obesity can induce PI1 expression in the liver and also continue increasing its level in the plasma. And this will serve as a compensatory stimuli to increase in TPA expression in the livers and also in the plasma. However, we also elucidate another mechanism that an increased dark one, which is a transcription repressor for TPA, to limit the further increase of TPA, which explains that the TPA smaller fold increase that cannot fully compensate the larger increase of pi1. And that leads to the eventual reduction of TPA activity and also reduce the fibrinolysis potential in the obesity conditions. So since we study obesity and dyslipidemia is also, or hypercholesterol and hypertriglyceridemia are also highly correlated with obesity. And if we look at the leading cause of death globally, both ischemic heart disease and stroke are the leading cause of death and contribute to a quarter of deaths globally. So we want to know that whether the mechanism that we observed also can contribute to understanding the heart disease and stroke. Because the heart attack and stroke actually share very similar pathology, because they both can start with the accumulation of lipid-rich plaque on the arterial wall, and they also both can be triggered by the formation of a black clot. So the larger question we want to answer is that are the black clot and lipid are correlate, and whether the having the TPA expressing in the hepatocyte only serve as another source of circulating TPA, or it actually serve as a link between the black clot and the lipid. So as TPA been used in the clinic for decades as a thrombolysis drug to dissolve the clot in patients that with cardiovascular disease. So there are actually lots of literature from clinical studies has previously indicated a negative correlation between the blood TPA activity with the high LDL cholesterol levels. So then we had another hypothesis, whether in addition to TPA's known function of breaking down the clot, whether TPA also can lower the LDL cholesterols. Now I want to briefly introduce the background of lipoprotein production in the hepatocyte. So all the ApoB-containing lipoproteins are considered as atherogenic, or also called as the bad cholesterol lipoproteins. So and all of those ApoB-containing lipoproteins have one copy of ApoB, and they are assembled mostly in the hepatocyte in the form as VLDL, or the very low density lipoproteins. Once they are secreted into the circulation, they can get hydrolyzed by releasing triglyceride and into the smaller size as IDL, and then eventually become the LDL size, where it can be uptake by the peripheral tissues and also eventually back to the hepatocyte for the clearance. And decades of research has developed to very efficiently lowering the LDL cholesterol by statins, and also in the recent decade, also using PCSK9 inhibitors. However, those drugs cannot efficiently lower the population cardiovascular risk, and the residual risk of cardiovascular disease are still high. So one of the hypotheses is that the statin and PCSK9 inhibitors cannot efficiently lower all of the ApoB-containing lipoproteins. So we saw that if TPA can reduce the VLDL assembly in the hepatocyte, and whether this can further lower the residual cardiovascular risk. So we first use a model to silence TPA in the hepatocyte in the atherosclerosis mouse model by feeding the Western diet to the LDL receptor deficient mice, and we observed a much larger atherol lesion in these mice compared to the controls. And if we look at the cholesterol distribution in the plasma fractions separated by the size exclusion FPLC, we see that not only they have higher cholesterol overall, but those cholesterol accumulated in the ApoB containing lipoprotein fractions which are atherogenic. There are two main mechanisms to determine how much VLDL will be secreted from the hepatocyte. One is how many particles are made, and second is how well each particles are made. To answer the question of how many, it's very easy. It's just determined by the ApoB concentration, because each particle have one copy of ApoB. However, to answer the more interesting question, how well each particle is made, we use the three approach. First, we measure the diameter of VLDL particles isolated by the ultracentrifugation, and then we measure the diameter using dynamic light scattering. And we saw after silencing TPA in the primary human hepatocyte, VLDL has a larger diameter. And then we also isolate the VLDL from the mouse plasma and using scanning EM. And we also observed that after silencing hepatocyte TPA, the VLDL particles are larger. Since VLDL, the most abundant lipid in VLDL is triglyceride. So we also can use the ratio of triglyceride to ApoB ratio as a surrogate for how well each particle are lipidated. So we also observed a higher triglyceride to ApoB ratio in the hepatocyte silenced mice. Since we observed that each ApoB VLDL particle carries more lipids after silence TPA, then our hypothesis is whether TPA can directly inhibit the lipidation of ApoB, which is mediated by the liposomal triglyceride transfer protein, or shortened MTP. And MTP is actually not only transfer triglyceride, it incorporates all the neutral lipids, including cholesterol, triglyceride, or phospholipid, into the nascent ApoB and start assemble the VLDL particles in the ER of the hepatocyte. And using a lipid transfer assay, we observed that TPA can reduce the lipid transfer activity from the donor vesicle to the acceptor vesicle using the fluorescently labeled lipid transfer tracing, but TPA can reduce this activity. We then isolate the ER fractions using density gradients, and because if the ApoB is more lipidated, then it should be accumulated in the lighter ER fraction because lipids are lighter than water. And indeed, after silencing TPA, we observed the ApoB is accumulated in the lighter fraction, indicating those ApoB are more lipidated after TPA silencing. Since TPA is a secretory protein, then we want to make sure that it really can interact with ApoB intracellularly, and more importantly, in the ER where ApoB are lipidated. So indeed, we use the confocal imaging and observed not only the two proteins interact with each other in the primary human hepatocyte, but they actually also correlate with the ER marker. And we also validate the two are in proximity using the proximity ligation assay. So what's the mechanism? TPA is known to have a unique lysine binding site on this Kringle 2 domain as the major mechanism of how TPA can dock onto the fibrin molecule that I introduced at the beginning of my talk. And ApoB also have the known surface exposed lysine residues on this N-terminus, where the MTP or the lipid scaffolding protein can incorporate the neutral lipids onto the nascent ApoB and start the lipidation process. So if we're just looking at the names, it's not hard to come up with a hypothesis that whether the TPA's lysine binding site on this Kringle 2 domain directly blocking the lipidation or the access of MTP to the N-terminus of ApoB. So we did many experiments to confirm this hypothesis, and this is just one of the examples that the blue line showing direct interaction between TPA and LDL or the delipidated ApoB. But once we use a TPA antibody that blocks the Kringle 2 domain, this interaction between TPA and ApoB will be blocked. So why our body designed this mechanism, having a serine protease which is a TPA to interact with ApoB in the hepatocyte. And as the main function of VLDL assembly and production is to redistribute the nutrients into the rest of the body. So what happens to this mechanism when we load lipids into the hepatocyte? And also, we have not talked about PI1 into this mechanism. And as the major inhibitor of TPA, which also expressed in the hepatocyte, was the role of PI1 in this ApoB lipidation process. So we use oleate, which is one of the most enriched fatty acids in our diet, to treat primary human hepatocyte to mimic the lipid loading condition. And we observed shortly after we load the oleate into the hepatocyte, we observed the free TPA is reduced at this molecular weight. However, there is a shift to the larger size of TPA, and we predict that the TPA and PI1 complex based on its molecular weight. And we confirmed that using ELISA with treatment of oleate, the TPA and PI1 complex are increased, and the PI1 free TPA is reduced. There is no TPA deficiency has reported in the humans. To validate if the mechanism we discovered is also happening in the humans, we collaborate with Dr. Amy Shapiro and her team in Indiana Bleeding Center, where they take care of the patients that have complete deficiency in PI1. And we collect the plasma from these patients and also compared their LDL cholesterol levels to the individuals from the same community, and also age, gender, BMI matched with the deficient patients. And we observed that the patients actually have lower LDL cholesterol. If we isolate the VLDL particles from these patients, and we also observed that they have more TPA associated with the VLDL particles, and the more the TPA on those VLDL particles, the smaller diameter of the particle is. So this is consistent with our hypothesis that TPA limits how much each VLDL particle will get lipidated. So this paper now is published in Science, and we showed that in addition to the known function of TPA in dissolving blood clot, it also reduces the LDL or the atherogenic lipoproteins. And in the basal condition, TPA binds to the ApoB N-terminus and block the lipidation process. However, after lipid loading, Pi1 forms a complex with the TPA and sequesters TPA away from the ApoB N-terminus so that the ApoB can be lipidated by the MTP. In summary, we found hepatocyte can synthesize TPA and contribute to about a half of TPA concentration in circulation. And obesity creates an imbalanced production of TPA and also its inhibitor Pi1 from the hepatocyte and contributes to the reduced TPA activity and also fibrinolysis. Within the hepatocyte, we also found TPA can reduce the apoB lipidation and VLDL production from the hepatocyte. And after finishing this study, Dr. Wen Dai also wrote a very nice review article summarizing the correlation between fibrinolysis and also lipoprotein metabolism, in addition to his own study, but also well-accumulated decades of study in this area. I encourage, if you're interested, you should check it out. And Mindy in our lab also wrote another review article more focusing on the VLDL assembly and also its correlation to the cardiovascular disease in general in another review article. And with that, I want to acknowledge everyone contributed to this study and also funding resources. And also, if you are interested in this topic, I will also encourage you to consider the Golden Conference that I will be chairing next year in Ventura, focusing on plasminogen activation and extracellular proteolysis. And I'm happy to take questions. Thank you.
So thank you, Dr. Zheng, for the insightful presentation. So we now have around 10 minutes for questions. I'd like to remind everyone that you can submit your questions using the Q&A feature if you have not done so. So we got a few questions. So our first question is the TPA always active? How is its activity regulated? Could you please?
Yeah, thank you for that question. It's a very good question. I did not have the time to show all of the study we have done, but it was a key question we want to ask. And we generated an enzymatic mutant TPA, and we found it still have the function of reducing the VLDL assembly to the similar level just as the wild-type TPA. So we believe that the enzymatic activity of TPA is not required for this function, which also makes sense because the protease domain is not the same with where the TPA interaction with the TPA, because the interaction with the TPA interaction with apoB is on TPA's Kringle2 domain, but not on the protease domain. So the protease domain is not required.
Thank you, Dr. Zheng. So our next question, is TPA bound to ApoE particles in serum? Or and the second question is TPA modifying the secretion of ApoE particles from hepatocytes?
Yeah, and thank you for this question. And I see the next question is also related to the following the first question. So I will just read the next question is, does interaction of TPA with ApoE particles can occur after secretion of both partners? So this is a really nice question. And we are currently working on this, and I actually don't have a definitive answer at the moment. And hopefully we can figure this out. And Dr. Wen Dai is also applying for his independent grant in this area to further study.
Okay. Thank you, Dr. Zheng. And our next question, how does TPA from blood serum enter a hepatocyte for function? I may have missed it, or is TPA expression also in hepatocyte?
Thank you for this question. At the beginning part, like the first 5 minutes, I talk about my earlier study showing that hepatocyte can have the endogenous TPA expression and also can be triggered by PI1, and everything occurs within the hepatocyte. So TPA does not need to enter hepatocyte to have the role of reducing ApoB lipidation.
Okay. The next question we have, what do you suppose is the role of LPA?
We are currently working on that. Hopefully can get it published soon.
Great. Thank you, Dr. Zheng. One more question. So does TPA interact with the cringe domains or LPA? It's a similar question, but
Yeah.
yeah.
Well, I don't think so. Because TPA has a Kringle domain and LP-little also have Kringle domain. So I don't have the hypothesis of two Kringle domain interact with each other.
Okay. Next question. Does ApoA has similar effects?
That's a very good question. I don't know. And I think it worth to study, and I think it is very possible.
Okay. So next question we have is any regulation of TPA on LPA?
Yeah, it's the same question we are currently working on. Hopefully can get it published soon.
Okay. So next question, have you looked at TPA knockout mouse?
If you are referring to the total TPA knockout, yes, we have looked at it, but there are trend of increasing apoB-lipoproteins in the circulation, but not consistently. And our interpretation is that there may have adaptation or compensatory pathway already picking up in those long-term knockout models.
Okay, thank you, Doctor. And we have two more. Do other cringe containing proteins have similar effects on ApoB lipidation?
Yes, very possible. And I see this question is from our also TPA and Kringle protein expert, and I think it's very possible, but just more study are needed.
Okay, thank you, Dr. So I think we have the final one. So do fatty acids with chain length and unsaturation structures that differ from oleic acid have shown the same results?
That's a very interesting question. We only tried oleate and also palmitate, and both have similar effects, but we have not tried the other fatty acid, and I think it's an interesting question to study.
Okay, I think that's all our questions. Thank you for everyone submitting such thoughtful questions. And Dr. Thank you for your detailed responses. So, Dr. I'd like to hand it back to you for any final thoughts or key takeaways before we close out today's session.
Yeah, again, I really appreciate everyone's question, and I found the questions are really in depth. And if you are interested in this topic, you should really come to our Golden Conference on plasminogen, and also consider submitting your studies to the Journal of Lipid Research.
Thank you so much, Dr. Zheng, for your time and expertise today, and to all of you joining us. Be sure to keep an eye out for the upcoming webinars in our Breakthrough webinar series, and we hope to see you soon again in October. And have a nice day, everyone. Bye.
Bye.